जी अस्सलाम वालेकुम, कैसे हैं आप सब? ठीक हैं जी. Today the topic that we have to be discussed is about the difference between a normal cell versus a malignant one. Actually, malignant, or we can say the cancer cell, we have to see that how a normal cell converts into a cancer cell. This is important. First of all, we must understand that to become a cancer cell, like just suppose if this is a normal cell and it is converted into a somehow a neoplastic growth, or you can say the over proliferations or becoming a cancer cell. So what could be the reason? First of all, to induce a normal into a cancer, there must be a mutation. There must be a mutation. So what is the concept of mutation? Moreover, most of the time people say mutation is merely a change in DNA. Change in DNA. But to be honest, to just change in DNA is not a mutation, because in a day or in a 24 hours our DNA is undergoing a lot of changes, but that changes could be or would be repaired. So such kind of changes are not regarded as mutations. So the mutation is true that there is a change in DNA, but what kind of change in DNA? That is non-repairable. That is non-repairable. So we can narrate in another way that permanent changes in DNA are called mutations. So this is the one major cause behind changing the genome of the DNA, the normal genome of the the normal genome of the cell into a abnormal, or you can say the cancer cell. Or actually there is a lot of damage to the DNA. We will see that how a normal cell convert into a malignant or cancer cell. But before it, we must understand there is a damage to DNA. There is damage to DNA. Now the point is that you must remember that it is not merely a single or twice damage to the DNA. It's a lot of the bunch of the damage. Understand? A lot of damage to the DNA. So what could be the reason for the damaging, or you can say the mutation? The reason could be viral. The reason could be radiations, high exposure to the radiations, or reason could be the, you can say, the chemicals, or the mutation also can be arise by spontaneously, like if there is no any history and DNA is undergoing automatic changes. Automatic means there is a mutations, of course. So but there is no any dispositions or depositions, or in the history there is no such a record in the progenitor histories and the parental history. So such kind of mutation is regarded as the spontaneously if these you can say the reasons are not there. The viral, the radiation or chemical mutagens are not there, so we may regard as the spontaneous mutation as well. So once we have the concept of mutation, we must understand that when a gene or when a DNA, of course there is a gene, functional genes or the structural genes, when genes undergo mutations, what can be done or what could be possible with the gene? So there is another concept, the concept of, just suppose if this is a DNA and just take this part of the DNA as one gene, this is another gene, and this is another. I am just showing the three genes, although there are about 25 to 30,000 genes present in human genome. Just suppose this gene is responsible for, you can say, the cell cycle, isn't it? And you can say this is for making hemoglobin, and this is just suppose this is the BCL gene. BCL genes are related to the apoptosis, etc. So just concentrate on the cell cycle gene. Cell cycle, okay, the genes related to the cell cycle, there could be the many genes, but just suppose this is the cell cycle or cell cycle proliferating gene. Mean whenever this gene is on, the cell undergoes the cell cycle. It will pass from the G1 and then S phase and then G2 and then mitosis or meiosis, whatever. So this is cell cycle proliferation genes. Now we will see that if the mutation is there, just suppose the gene is altered. The gene is altered due to the mutation. Then what could be the outcome? That what will be happen with this particular gene? Of course, the gene is making to the messenger RNA, and then messenger RNA is translated into the protein. And of course, if there is a mutation over there, the messenger RNA will be altered, and if the messenger RNA will be altered, of course the protein will be abnormal. So what could be the two possibilities? Actually, whenever the mutation is there could be the two possibilities about the gene, that either the gene gaining the function, gain of function we say, or loss of function. This is the same happening in the society, that if a person is doing his best, and by some reason it got mutated by irritations or by any lesson, so there could be the more two outcomes. Either the person become more good, or either the person will become bad, or leaving that particular roles that he is already you can say play. So the gene has two main possibilities after the mutation, either the gain of function or loss of function. Gain of function means if this particular gene, cell cycle proliferation gene, is getting the mutations in such a way that the gene is getting the gain of function. So what could be the possible? The cell undergoes not undergo the normal cell cycle, but over proliferate. This is the one implication. And just suppose if this particular gene, the cell cycle proliferation genes, mutated in such a way that it gets the loss of function, so it will never proliferate. So this is the basic difference of the mutations. Whenever the mutation is there, you must have to be understand that either the gene have gained the function or have lost the function. Then you can understand well that what could be the outcomes or what could be the implications. So up to now, if you have understanding about the mutation, so now we can understand best about the normal cell into a malignant cell, how it will convert into the malignant cell. So let's start. Just put over there, just suppose there is a normal cell and there is the malignant cell. The basic difference I am telling you, inshallah in future I have a full lecture plan. Okay, so just suppose if this is a skin piece, isn't it? And anyhow, if my skin got damaged, first I am telling you the normal thing. If the skin undergoes damage or any injury, so of course there will be inflammation over there. inflammation and vascular events and different, and then the cell underlies over there, they start proliferating under the particular signals. So they start proliferation, and they will proliferate and fill up the gap, fill up this gap, and then stop. The normal cell knows that when to proliferate, when to proliferate, and how much to proliferate, and then they stop. This is normal. It's mean the cell cycle, or you can say the normal cell cycle, is under the very strict control. Cell is not, you can say, the self sufficient in its growth. Either it is under the particular signals, and it is obliged to, you can say, the follow the rules to grow or to proliferate in particular manner. In a nutshell, we can say the normal cell never grows on its own self. Either it has to grow, it is only grow when there is a need, and it will stop. It's mean it will obeying the inhibitory signal. You can say it will obey what? Growth signals, growth signals, as well as inhibitory signals, as well as inhibitory signals. So that's why when there is a growth signal, it will start proliferating, and when there is inhibitory signal, it will stop proliferating. This is the normal cell behavior. Now come to this point, malignant cell. Malignant cell is such a, you can say, a nasty cell that although there could be a possibility, of course, okay, for cell cycle there could be the genes. One are the, you can say, okay, let me draw the genes. Rather, I think we should start this discussion in the part 2 lecture. So up to now we must understand the core difference of the mutations, the gain and function and the loss of function, and then we have to start the lecture in the cell cycle. So the first difference is the cell cycle. There are a lot of things. Actually, the most of the students knows the to become a cancer cell from a normal cell, there must be a somehow hera-pheri or damage to the cell cycle only. But this is the one aspect. This is not the whole story. So we have start the cell cycle story first, so we can understand that how cell cycle undergoes. First of all, we will see a normal cell cycle, how a normal cell cycle operates, and then what could be the you can say damage, and what could be the implications. We will see in the next lecture, in the next part, part 2. Take care of yourself. Allah Hafiz.